Thank you all for coming to the second iteration of our seminar series, the Science of Teaching seminar series. We're really excited to have everyone here. And we're really excited to welcome today's speaker, Dr. Utpal Banerjee. Dr. Banerjee received his PhD in chemistry at Caltech and also did his postdoctoral work there in Seymour Benzer's lab, where he started to develop his interest in Drosophila development. Dr. Banerjee is currently the Irving and Jean Stone Chair and Professor of Molecular, Cellular, and Developmental Biology at UCLA. And among his many awards, he's been named by UCLA Today as one of the top 20 professors of the Bruin century. He's received the Elizabeth W. Jones Award from the Genetic Society of America, and he is also currently a Howard Hughes Medical Institute professor. And Dr. Banerjee is really a pioneer and innovator in undergraduate education, and that's really the reason that we brought him here today, because he's helped develop multiple discovery-based laboratory classes that allow students to be involved in original research. And these classes have led to the publication of multiple journal articles that include hundreds of undergraduates as authors. And he's also helped to create a course that helps students learn how to interpret and understand a scientific presentation from some of the countries in the world's top scientists. And today, Dr. Banerjee, in his talk, Construction and Deconstruction, a comprehensive research program for early undergraduates at UCLA, is going to tell us a little bit about these different types of classes he's developed. So will you join me in welcoming Dr. Banerjee.
Thank you, Evan, very much for the kind invitation and introduction. I also want to mention that I brought my colleague with me, Dr. Ira Clark. Everything that I talk about here more or less was developed along with Ira, and he currently serves as a sort of an organizer and co-director of the biomedical research minor that I will talk about in a few minutes. This is not showing the whole slide. I hope that won't be a problem. There's a whole line missing below this, which says, with Ira Clark, John Olson, Rafael Romero, and Corey Evans. These are the four heroes of our program. So this title, Construction and Deconstruction, where does that come from? These are all homegrown ideas that we've come up with. I'm not an educationist who's going to tell you a lot about how to educate people. I'm just simply going to be able to give you a report on what is it that we have done, which we feel is useful for the undergraduate student as part of their curriculum. The idea of construction is to take large number of simple observations and maybe make something which is much bigger with it. So sort of like using these Lego sets, which can be combined together, and if properly combined, could build something really magnificent. So you start with minimal elements. Large numbers of small observations is the critical key to this. The opposite is the idea of deconstruction, which is you start with something that is as magnificent as this, perhaps like a research work that is going on in somebody's laboratory, and then now you break it down, not so much to the very individual Lego piece, but to pieces like this, such as the minaret or the dome or the corridor and so on and so forth, such that one can then, after breaking this down, show the student how one can take each of these larger pieces and put the whole structure back together. So the two goals that Ira, myself, and others have is how can we best prepare our students for careers in science, and can we make research a core part of the science curriculum? In other words, can early engagement of scientific inquiry improve science education. So the usual ideas that most students have is that they have to finish reading up every possible textbook and every possible course till they go into a research laboratory. And we tried to come up with this experiment maybe about 10 years, almost 12 years ago now, to say that let's just put the student in front of the microscope. That was one of the biggest challenges when we proposed this class to UCLA. A certain person who was a professor in English said, "Are you sure your students will know how to focus a microscope because they're freshly out of high school?" And I said, "Well, we'll make sure they know how to focus the microscope." So then, should they be able to do really well in their didactic classes? There's no direct proof that it works that way. It's just that there is a lot of indirect evidence to suggest that these methods have, in fact, shown a lot of results. So people have published publications, and if you want to look up, I suggest there are these 3 pieces of publication that people have talked about. Vision and Change by AAAS, which talks about applying the process of science. And we really like the Vision and Change, not just because they talk about our programs quite a bit, but also because it's a thoughtfully written monograph on what is it that one wants to do, what wants to achieve and see in undergraduate education. The second one is this HHMI's AAMC competency idea, that what are the basic competencies that a student should be given in order for them to get into a medical school. So this is a bit medical school oriented, but nevertheless, this idea of whether or not they're getting competency in various subjects rather than being taught very large amounts of each unit that they are participating in is something that is worth looking into. And then finally, this precast or the Presidential Council of Advisors on Science and Technologies Engaged to Excel. It's something that Obama had requested, and then a good friend of ours, Joe Handelsman, who is right now involved in the White House for developing programs, has suggested over a long period of time. And the main idea is STEM education and graduation rates have to be improved in the entire country. 2 of the reasons that are often quoted for dropout from STEM, number 1 is deficiency in mathematics, and number 2, boredom in their science classes. The first one is being handled by various groups right now at UCLA. We are not involved with the math education in the program that we talk about, but we hope that we make our science a little more interesting in order to retain our students. So this biomedical research program at UCLA that we have developed together has 3 different components to it. One is called URCFG. This is the worst acronym that one can come up with in the entire world. It's difficult to remember what it means, but I think it's Undergraduate Research Consortium in Functional Genomics. Now, one really nice thing about this nice name is that it gets the undergraduate hooked right on the first day. Because on the very first day, I tell them that, look, without even lifting a pencil, you have now participated in the Undergraduate Research Consortium in Functional Genomics. How good this will sound on your CV. So this can take about 60 students per year, between 20 to 30 students per quarter. So we can run up to 60 to 90 students in this program. And then this research deconstruction, which is for 300 or so students per year. And these introductory programs are given to first and second year students. We like to restrict these to people who are freshly off of high school if we can. The only reason we allow a few third-year students is because we have transfer students, and these guys have had absolutely no ability before to do what we are trying to present to them. So if someone is a transfer student, we try to make an exception for them because they're usually very driven. And then from these two programs, we take a group of students who then become part of this minor in biomedical research. These programs have trained over 2,200 students in inquiry-based, research-based programs since the year 2003. So we've been running them for 10 years now. So now to take you through these programs one at a time, the URCFG, or the hands-on research. As I said, this is a mouthful, and that helps us. This is the one that is actually sponsored by the HHMI. We took about a quarter just to draw a picture of a fly such that the abdomen looks kind of like a DNA, and still couldn't get the veins right on the wings, I guess. But nevertheless, this is kind of our logo here. And 771 students. So one thinks, what change are you going to make by 20 to 30 students a quarter? Well, eventually, if you have lived long enough, you will make a lot of changes to a lot of people. Oh, I can see Gloria Young right here smiling away. It makes sense that she has a bigger picture than anyone else. She has been a fantastic student, a TA for the class, and also a co-author on a Cell paper that we just published. So, but nevertheless, a lot of these people, this is Yuki, it's a large number of people, but it still is small enough, and it's small enough at a time that we can in fact know these people, we know what they're doing, and certainly John Olson, who does the lab part of this class, knows them very, very well. So what do we do with these students? And of course, this being a UC system, everything has to get a course number. And so we have called this LS10H, Research Training in Genes, Genetics, and Genomics. It's entirely voluntary. It's not required by any department, and any department students can take it. We have had students from the history department, of course, MCDB and other related fields, but any department students can take this. And the idea here is to have one central difficult concept, difficult that is for the student who is just starting off beyond his or her high school days. It has a multifaceted approach, lab, lectures, computer, and writing. I guess it's better to describe it as a boot camp rather than a class. Within this 10-week period, they get a lot of different things to do. Simple approaches, true functional genomics, but simple approaches. And because my lab works with Drosophila genetics primarily, therefore we choose programs which are usually all Drosophila genetics based, compiling large number of small observations, as I mentioned to you a few minutes ago. So in the didactic part, and that's where I give a few lectures, and Olson gives the rest of the lectures. We not only talk about background material, but also about ethics, career options, grant manuscript writing. In fact, their midterm is writing an NIH-style grant proposal. They have to do that for the rest of their lives anyway, so might as well get started early. And their final exam is writing a manuscript. Any format is okay, but somehow they all seem to choose Cell. Then some amount of bioinformatics, record keeping, laboratory fly pushing. They all have a notebook in which they how to do all this. And they write at the end, they will write a really nice sort of a paper, a proposal. So these are the three major projects that have been undertaken by URCFG. mosaic analysis of essential genes during eye development. So this is being completed. So this is it's not so hard for Drosophila people to appreciate what is being done here. It's a simple case of mitotic recombination in the eye for genes which are otherwise lethal. Very, very hard for the undergraduate to appreciate on the very first day, because if there's one thing that has been drilled into their brains since day one is that mitosis and recombination don't go together. So therefore, just to get them intrigued and get them thinking about how one can have mitotic recombination itself is an interesting process. Now, in this, they have taken 2,000 lethal mutations in Drosophila, made clones in the eye, and then studied what their phenotypes are. The second is a gene-based lineage analysis. This has also been completed. 700 genes that have been studied for development in brain, blood, eye, wing of Drosophila. And then this is what they're doing now, an RNAi screen for controlling hematopoiesis and cardiac development. 5,000 mutations are being studied this way. To give you an idea of how much work is involved in each of these projects, this is project 1. Took about 2.5 years to complete. About 3,000 separate introductory experiments performed, greater than 120,000 Drosophila crosses performed, 2,000 mutant stocks, 1,375 published stocks, 1,576 verified phenotypes, and then excisions to show that they are real. So this is a tremendous amount of work which I don't think anyone will want to take on in anyone's own laboratory unless one is crazy. And the idea is that you just simply give each person a whole bunch of these lines which have got lethal mutations on it, and they become homozygous in the eye. And when that happens, you end up with all kinds of mutant phenotypes, which they then create databases for. And then the real part of the work is the analysis of the data. They go back and look at the genes, what the functions might be, and why they got picked up in their screen. That's what they write in their discussion sections. The second one had to do with creating this GAL4 expressing line, which causes lineage tracing. This is for people who work on mouse here. This is as if you were doing a Rosa mouse experiment, except with every gene that is there in the genome made into one of these Krelok's excision systems. So you can then create a situation where with just looking at GFP and RFP, the cells which are currently expressing a certain gene and the cells which used to express that gene. This is a lineage tracking which can be done by just having a GFP-RFP scope and being able to do these dissections. And they have done that for a very large number of genes. And the third one is to take these fluorescent flies, larvae that we have generated, such that only the blood system is highlighted here, the circulating cells and the heart and the blood. And then if the heart and the blood becomes more or less or melanotic tumors form, then they can score those for different RNAi lines. A lot of work. This is just taking the labels on their vials, and then putting one on top of the other. This is kind of a statement. Now this has degenerated. They have started putting years on it and making it into a Mickey Mouse. So I've stopped photographing it. But it's a huge amount. This is a solid ball of labels up to that height. Okay, we also have various high school programs. One school that we had adopted early on is Loyola High School, simply because a teacher from Loyola High School was sincere enough to come and take this class with us at UCLA, then go back to his lab and then have them do this, not in the larva, but in the adult fly. And this is John Olson, our person who teaches at UCLA. He then visits Loyola High School, which I have done as well, and then talk to them there. But our larger program is where during the summer these students come, and then this is the high school outreach. And about 10 to 12 students, sometimes 15, about 70 students have been trained with the same program that the undergraduates, and they seem to do well in terms of where they want to go. So everything needs to have some kind of an output that says that a certain amount of work was done. And I know that the postdocs invited me here, so one of the ways in which I want to emphasize here is that one does not need to give up all research and just concentrate on teaching students in order to make this worthwhile, that in fact one will embellish and help the other. So this was our first paper in PLOS Biology with 134 students, undergraduate students included as authors. And then this was beaten by our genetics paper in 2007, 264 undergraduate authors. I believe still this is still the record of the number of undergraduate students on any publication, but we have now another paper which is supposed to go out pretty soon, which beats this number by a little bit, 300 something. Okay, so there are also various tools and techniques that come out. For example, this G-trace technique, other than Corey Evans, John Olson, and here, obviously, Volker Hartenstein, Gerald Call, and myself. Everybody else here is an undergraduate student. And together, they developed this method for lineage tracing, which came out in Nature Methods. For our assessment purposes, there are many different things that are done. The usual UCLA assessment is made, but we also send out all the data to Grinnell College, which does independent assessments in a blind fashion. And you can see the triangles represent the Grinnell College assessments compared to the various programs. And I think they have got tens of thousands of students included in their nationwide programs, which serves as controls. And so in many different categories, this is a self-reported system, so it has its problems, but at least we are doing okay. Now, this is the slide that I'm most proud of, and Ira did a lot of work with other people in UCLA to dig these numbers out. For our underrepresented minorities within this program, the STEM retention has been 100% in 10 years' time. For the overall program, it's 95.2%, 96% of women. And this is not just a flash in the pan one program. Of course, this is all 771, not quite this quarter, but many, 750 students put together over the last 10 years. So it will average out any kind of irregularities in the system. For comparison, national average is 35.1. UCLA average is considerably better, 69.3. But UCLA underrepresented minority is not good, 48.8. We still do not have as many URM students as we should, and one of our goals over the next year or two is really to increase the number of URMs, but the retention is quite nice. This is only the first program that we have gotten the retention numbers on. We are in the process of working it out for the other stuff. Okay, so what is research deconstruction? This was just some figment of our imagination, thinking that won't it be good if one were to listen to how an experiment is done first and then to be explained. See, everyone does that. The English professor doesn't first come and tell the students, what Shakespeare or Byron really meant in their ballad or poem. They first read the ballad or the poem, then there is an analysis done of what is it that they actually read. We, on the other hand, as scientists, as technically competent and critical as we are, we go and tell them, this is exactly how cell division works, this is exactly how the cytoplasm is supposed to react under this condition, that condition, the membrane. Sometimes we tell them it is fluid, sometimes we tell them the organelles are like phase transitions. We just tell them what is it supposed to be, instead of first giving an example of how we know what we know, and then try to explain to them. And we thought that if we took that approach with starting students, won't it be good? And we have in fact tried this even with general education, and it seems to work. We have now done this for a long period of time. So what is it that we do? Oh, by the way, this deconstructing scientific tool, etc., is published in PLOS Biology. And again, it was mentioned in Vision and Change as one of the many bona fide methods for achieving this inquiry-based learning. So what is it that we do? Up to 90 students, mostly first and second year, and we would first have a full-scale research seminar. This is by an invited faculty member who can talk about, we tell them not to dilute it down. We like it to be full of data. Philosophical talks are not very good to deconstruct. If there's a lot of data in it, that really is good for us. So they give this full-scale research seminar. As you can imagine, we have an eminent scientist coming and talking to a group of students who are just off of high school. And so everything that he or she says, they nod, but it kind of goes above their ability to comprehend. But largely, they pay a lot of attention because they really want to know what is it that this person is telling them. We then videotape this seminar, and then we place it on a website with copy of everything for the students to review if they wish. And then we have these deconstruction classes, and this is the critical part of the whole program, which is that we then have a lecturer or a professor who comes in and takes that seminar as if that seminar was a textbook. And every 10-minute segment of that seminar is then analyzed and deconstructed, so to speak, into little pieces for the fundamental that one was trying to achieve. Of importance is, what is it that the scientist who was doing this experiment was thinking, was doing. Scientists, when they give talks, have a way of giving talks as a storytelling. That's a method that we know works really well for expert learning. And I want to make this difference between expert learning and what is going on here. Many people would ask that if discussions are so important, then why are you giving this talk in a lecture format here? Because this is expert learning where one is already primed to listen to somebody and they are all equals in terms of what the content is. This is not the same as a person trying to learn how to do research who has had no experience doing research before. So these deconstruction classes are done over a 5-week period. So say 1.5 hours lectures. So that would be maybe 10-15 hours worth of lectures that are given to them. And then a question-answer session, which is very important. The original speaker is invited back at the end of the series. And it's amazing to watch that these same students who were quietly listening to this person on the first day then become very verbal and they start talking about it. Because in the meantime, they have listened to a deconstruction of all the lecture, but they've also read various things that this person has done and related studies on which they have been quizzed and tested and so on and so forth. Okay, the nice thing is the reason we developed this is because it's easy to export it. Not every place will have the resources needed to have the hands-on experience. It's scalable, so we are no longer limited to the 15 to 20 students. We can do it now with 100 students. It's adaptable to multiple research disciplines, so it does not have to be based solely on Drosophila genetics, which is what I understand and therefore I'm limited to. But this can be done for anybody else, and it works in Hebrew because Benny Shiloh tried it and tries it every year at Weizmann Institute with a lot of good results. So here is an example, and I'll show you several clips of this. So here is Ben-Hur Lee, one of our premier virologists at UCLA, who will talk about the Nipah virus. And then right after that, Rafael Romero, one of our instructors, will take a small part of what Ben-Hur said and will try to deconstruct it that makes it understandable to the undergraduate.
As you can see, the codon-optimized protein gives you many more syncytia. This is just how you quantify syncytia than the wild-type sequences. And so this is just, I know you can see it, but it's real, and you just have to be used to seeing these kinds of syncytia. When you transfect the codon-optimized F and G glycoproteins, you get many more syncytia formation than the wild-type version, and you need to basically codon-optimize both F and G. One by itself is not sufficient to give you the kind of phenotype that you want to analyze. So you're going to have some Gs, you're going to have some Fs and some Gs. They're going to be on the surface of the cell. All right. So in other words, this cell is making viral proteins. You force the cell to do this, but you need it to do this experiment. And it's another way of working with these proteins without working with the virus itself. All right. So in other words, when G connects to ephrin right here, or G connects to ephrin right here, these two will actually stick. They will attach, just like a virus would. And when it attaches, because you've got the F protein, remember the F was essential for fusion, these will actually fuse like they were a virus in a cell. And so what do you call this right here? We call this a syncytia.
Okay. Then the point is that you can do this with large numbers of students, but its efficacy goes up if the number of students is smaller. And so if you have a few students who are at a more advanced stage of learning, where they have already taken the first couple of classes, then we bring in some experts to turn the knob up a little bit more, and then we do this, what's known as the enhanced deconstruction class. This is something that Ira teaches here, and this is there's a little bit more of primary literature reading, student presentation, strong writing component. The reason why we tried this was because, remember that Lopato assessment scheme, we found that there were many places, and I'll show you this, where the students do well as the hands-on experience. But there are other things that we are not concentrating on, such as student presentations and strong writing components, and there it automatically drops. And so is it possible to use this method to get a higher output? And it is. So here's an example of the kind of a little bit more complex thing that would be done, which is we brought in Tim Lane to talk about Wnt signaling in breast cancer. So this is a very complicated sounding paper, which about which Tim gives a talk. And then in the deconstruction of it, they are learning a lot of different things. So it's not as much of a lack of breadth and depth only as one would think. There is the cancer biology, the signal transduction pathways, what is Wnt pathway, cell cycles, ubiquitin mediated, and so on and so forth. Most importantly, though, the techniques, transgenic mice, immunostaining, so what kind of controls will you use, RT-PCR, what kind of reference RNAs will you use, what kind of loading controls will you use? And it makes a big difference, as all of you are educators, so I don't have to emphasize this, that looking at a gel in a paper versus saying, what did the person actually do? They took this, they ground it up, they ran this, then they used an antibody, and so on and so forth. It completely changes the perspective with which the individual student looks at that gel again. So here is Catherine Plath, who works on iPS cells, and this is from one of the enhanced deconstruction things.
If we add G418, which is the drug, into the media, and cells don't express this neomycin gene, then the cells will die. So, but if this resistance marker is expressed, then the cells will survive in this drug media. So basically, his way of selecting for a reprogrammed colony was he puts in these 4 factors into these cells, and then he puts the drug selecting for expression of this promoter into the media, and just waits if anything grows out. And he got something, right, and that's what he called induced pluripotent stem cell.
You're getting at what was actually used and what ends up being a very useful strategy here. So Lon is remembering that in Dr. Plath's seminar, they used drug resistance to identify the rare cells that are expressing the embryonic stem cell genes. You guys have all seen reporter genes. Right? Those of you who were in 5HA saw reporter genes in Dr. Arispe's work, right? Where she had VEGF-LAC-Z, she had the promoter of VEGF driving LAC-Z, so you could see in the animal which cells were expressing the VEGF gene. Those of you from LS10H used reporter genes because, remember, you had all these GAL4 lines, and each one of those GAL4 lines was turned on, was expressing GAL4 in different tissues in the fly, different cells in the fly, and that was because GAL4 had been inserted next to the promoter and enhancer of a nearby gene that was active in those cells. The point is that when this promoter is active, when this gene is expressed, now instead of FBOX15 being made, you'll make whatever reporter gene you put in there. Right? You'll turn on the gene for neomycin resistance. And so that's what the Yamanaka group used as their assay. So are the fibroblasts going to be resistant or sensitive to neomycin? Fibroblasts should be sensitive because this gene should be off. Now if we get that rare cell that's behaving like an embryonic stem cell, then what would we expect? Would the cell be resistant or sensitive? Should be resistant. So that's what we're looking for.
I'm sorry, he's not answering his own questions. We have a very primitive recording device where we cannot hear. The students are answering, and then Tanara is going back and reinforcing the answer. So in terms of the diversity, we take so many different professors from UCLA, everyone that you will recognize in your own field. So all kinds of different people will talk about different things. It's a hard thing to teach because every quarter you have to, the person who is teaching it has to learn a new field in order to teach the undergraduate students. Now that's a little difficult, but of course that's why we have people like Ira. So as I said, the competency goes up in various things, not in every aspect for which we have the enhanced deconstruction. Now, we don't want to be left behind in this sort of online activities and education and so on and so forth. We don't believe that's the only way to teach an undergraduate student. But what we did was to show that you can actually do deconstruction of something which is online. So here is David Morgan from UCSF, and Ron Vale has made a large number of online lectures by various professors across the country. We took that online version, we deconstructed it, and then we called him back by Skype and had him answer questions. And the whole process is now being videotaped thanks to HHMI. And we are going to put that on our web as a website, so anyone who wants to see what kind of activities, what other things they did, they'll be able to do that. So here's the online, and you can see that the quality of video is much improved.
So for example, N6-benzyl ATP can be used by the analog-sensitive CDK1 kinase, but cannot be used by a wild-type kinase because that bulky ATP analog can't fit into the wild-type active site. And so, of course, if you put a radial label on the gamma phosphate of this bulky ATP analog and then add this kinase to a crude cell lysate, what you hope to get is the specific labeling of just the direct targets of that protein kinase and no other kinases in the cell lysate, because those other kinases can't use this bulky ATP analog.
So this is the iBio seminar taken from the web, and then we can get this one deconstructed. I won't play all of it because I'm running out of time.
What's the flaw of the experiment that I'm proposing right here?
If there are any other kinases and their substrates in the tube, then those substrates will also be phosphorylated.
Right, and where are those other kinases going to come?
From the cell lysate.
Right, so you'd imagine that this cell lysate is going to have other kinases. Sure. I mean, there are many different types of kinases in cells. And kinases use ATP to phosphorylate. So what ATP are they going to be using in my particular test?
Okay, so you get the idea. And here is David Morgan, then called back from his office at UCSF.
What else did we talk about? Here's one.
Hi. When running the mass spec, you attempted to find substrates of CDK1 in an unbiased fashion, but then at the end of the experiment, you identified the substrates via a consensus sequence, correct? So do you find in a way that biases the experiment itself, and do you think that you miss substrates based on that?
Yeah, that's a great question. Basically, there's this long history in the field of CDKs that there is no known substrate, real bona fide.
Okay, sorry to cut you off, but you get the point that they start talking a lot more when they have already gone through the deconstruction classes. Okay, a small number of students from each of these programs is then taken into our minor in biomedical research. This is a minor which now has almost 200 students. And this is the idea. They have already been recruited early. So now we place them into laboratories, extensive research commitment, at least 4 quarters, but much longer, usually for 3 years normally. They get research training courses, and they get some courses in the social sciences, either a history or philosophy of science or a bioethics class, which they have to take in order to fulfill the minor. And then at the end, they have to write a thesis. Now, minor was launched in 2007. It's provided research training to 340 students in 147 laboratories at UCLA from 30 different departments. I mean, there are some advantages of being in a place for 20-something years, 25 years. You know everyone, so when you tell them to please take your student into their class, and they have been trained already, and they have been handpicked, nobody usually says no. It's very easy to find people, particularly in the medical school, to take these students in. 195 students are in 95 different laboratories in the School of Medicine. Now, expert learning. At this point then, these students are starting to become experts. They have already gotten a certain amount of background. At this point, we start bringing them in at 18 students or so together, and they discuss what is it that they are doing in their laboratories. with each other present and with 2 of the instructors present. This has many purposes. So people often ask what's the difference between a journal club and the deconstruction process. And this is the difference. Here, this is more similar to a journal club. Now they are coming in, they're talking about their own work, they are quite an expert in their own work. The advantage of this is they are making these presentations with the other students present there. So if they have been washing glassware and the other person is about to publish a paper in Nature, they know the difference. We don't have to tell them that. This is very clear, having to make these presentations in front of your peers. You know, we are always petrified doing it. They are petrified doing it too. So as a result, the learning goes up enormously. And this is something that Corey Evans does this. And to define the research topic, to what are the messages, how should they have the slides, personal style, oral presentation, critical questions and ideas, and so on and so forth. This is Corey's experience with the students and expertise, I'm sorry. Now, the thing is that these minors, we have started in 2007, they have published 90 papers. This does not include my papers that we talked about with multiple 300 authors and so on and so forth, not including those. These are students who have gone to labs, including our own lab, but to many different labs, and then they have published papers from there. And there are 90 publications from these students in pretty good journals, and 35% of all the students have at least a publication. And we are trying to hope that this will even go up, but this is quite a good number of papers. Ira, with a lot of work, had made a list like this of all 90. That would have taken about 3 or 4 minutes to show, so with apologies to him, I took that out. Here is his first page, and of course, I'm so grateful that our recent Cell paper got highlighted as the first one. This is Ira's doing. Thank you. And then there are various other. So, but these 2 students, Gloria and Denali, they didn't just get on there. They did a lot of work. This paper talks about olfactory control, and Gloria actually took all 25 olfactory receptors, knocked them out, and looked at the blood phenotype and so on and so forth. So it's a lot of work that these students put in, and there are 90 papers like that. After 2 gap years, because that's the fashion these days, students always want to take 1 or 2 gap years. During that gap year, they do great things. They go to Cold Spring Harbor and work there, or they come to UCSD and work there, and they do fantastic things. But after that, 84% of our students that we have tracked, in fact, go on to graduate degrees. 45% MDs expected, but a significant number of PhDs of either MD, PhD, or PhD kind, 26 plus 18%. That's a significant number of students who then are going into research-based graduate programs after finishing this. And of course, one hopes that the MS and so on and so forth will also join in to that group. They go to good places. So that's and they get a lot of different kinds of awards. Now, I'm going to take exactly 2 minutes, because that's 2 or 3 minutes, to make this one kind of appeal mainly for, again, the postdocs that have invited me here, which is to say that I don't think spending this much time with education programs and developing these programs hurts anybody's careers. Okay, this is a common misunderstanding, which has been complicated, of course, by our tenure committees and so on and so forth, not giving proper importance to education. But if one plays one's cards right, just don't go die for a cause. There is a good way to get a lot out of this whole thing as well. I know I'm sounding a little cynical here, but it does make, you can benefit a lot from involving the undergraduates, and the undergraduates benefit a lot from their involvement in these programs. So here is the third program of our LS10 that we were talking about, which is a screen for various kinds of blood mutations in flies. Now, what we did was to take all of the positives that the students got from the screen, and Bama, who is a postdoc in my lab, then did a little bit more fancy analysis on them. He has spent about 4 or 5 months on it. So here are the blood cells in Drosophila, circulating hemocytes, and then they look for all these different phenotypes. This is what the students do, along with all the other stuff that I mentioned doing. Now, why am I showing this? 665 of those 5,000 lines that have been tested were considered positives by the students. Now, when my postdoc repeats it, 215 are real. Okay, so please do not go and start sending papers out with raw data that you get from the classes. Get them validated. It's not because the students are not paying attention or making things up. They truly see those as phenotypes. It could be a chunk of food stuck to the cuticle, for all you know. But on the other hand, they see that as a phenotype, they photograph it, but only 1/3 of them are real. But nevertheless, that's a lot of genes, 182 genes corresponding to those 215 mutations. So these are all the genes. Anyone wants to work on any Drosophila gene? I can give lists of them for people to work on. Now, if you do an analysis of these genes, you find various clusters come up of who interacts with whom. And if you take, say, just one group of genes, the CSNs, which are the signalosomes, which are very important for cell cycle, one finds that in this 3 to 4 year period, by having people working in groups of 30, 15 or 30, they don't know each other, they got random genes assigned to them, this is what they picked up. CSN1B, 4, 8, 7, 5, 3, 6, and this one is called alien in Drosophila. These all interact with each other. They form the COP9 complex, which is very important for a whole variety of reasons, but their role in stem or progenitor cell maintenance is just now being figured out. And so one can follow this up and look at what happens to the circulating cells. They can be less. There can be these gigantic cells forming. There can be these what are known as lamellocytes forming. These cells, I don't even know what they are. It's just open there. These are these binucleate cells which need to be analyzed. So the point is that there's a huge trough of data that can come out of these programs, which can then be analyzed at much greater detail for its research importance as well. So those of you that are planning to go to small schools to use undergraduates, go for it. NIH will support smaller research programs like that for doing something that generates a large amount of data. I have to thank Ira Clark, John Olson, Rafael Romero, and Corey Evans, the 4 people who are the instructors, and they have various titles in addition to being instructors. We have a half-time person who helps us with administration. And we thank all the deans, and these are the advisory committee members. And there are 145 UCLA faculty members. Thank you very much.
We have time for a few questions.
For me?
Yes.
Okay. Hi. So we have a research project that we have in the undergraduate labs here as well, which I'll talk to you more about at the roundtable. But my question addresses the quality control issue that you brought up a few minutes ago. And so if students are identifying things incorrectly or entering data, in our case, incorrectly into databases, do you then go back and have somebody verify all of the information before you publish?
Everything, yes. You wouldn't publish it in genetics or PLOS biology with mistakes in it. So we have, but it's a matter of having a system worked out. Initially, one thing that we used to do was there are these students who, instead of wanting to go to other laboratories as part of our minor, there are a group of students who want to just stay on to doing what they were doing, particularly during summers. Okay, we call them lifers. And these lifers just don't want to leave. Okay, we have kicked them out, we have gotten them to the next step, but they just want to stay with this one little LS10H program. They then become tutors to the next generation, and then they also become so proficient that during one summer, they could probably repeat everything that is been done for a whole year or maybe 2 years' time. What we can't do is to repeat every experiment. So if some experiment has had a negative result, that ultimately gets lost. And we just have to live with that if someone had a brilliant mutant but didn't score it, that's probably lost forever. But if someone says they have a positive result, which is what we will publish, then we have that repeated again. Now, having said that, each of these lines that they are screening in the class, so for the latest one, we give them about 30 to 40 of these lines to screen, each student over the 10-week period, 30 to 40. It's mixed in such a way that over different quarters or within the same quarter, 10 of them are completely unique, and 10 of them may have been used in previous quarters, 10 of them have been used and scored as positives. So we have an internal control over the quarters as well, in order to see that the same thing gets checked twice. But in spite of getting checked twice, there's still problems. And so they have to be resolved either with summer students, or in this case, I just thought, okay, this postdoc of mine is fantastic. So for him to recheck everything took only a few months.
Hi, I'm completely blown away by your data on retention rates in graduate school, and I've been involved in some interventions, and we don't even come close. But one of the questions that has been raised when I've tried to talk about the value of these interventions is self-selection bias.
Yes.
Meaning that, well, the students who went to whatever activity were the good students. And it's been very difficult for me to try to find ways of addressing that. And I wonder if you have any thoughts on that.
Yes, I mean, that's a very common, of course, particularly for us. We have no problem saying that we are taking motivated students, and the goal of our program is to take motivated students and make them outstanding and give them the best that UCLA can offer. So that's point number one. Point number two is, yes, there's a self-selection bias, but if self-selection was the only reason for URM dropouts, then I think some of the really strong schools that have got extremely high GPA requirements, extremely high selective advantage over other schools. I'm not naming such schools in the East Coast that charge very high amounts of tuition, would not have an URM problem ever. Yeah, because look, they got the best of the best of the best from the whole country. And so why should there be any STEM retention problem? If one looks at those schools, one finds that they face, and they will be the first ones to agree that they face as much of a STEM retention problem as a public university does. And so STEM retention is not just a matter of having the good group of students. Of course, that is one of the aspects. But it is also a matter of having a positive input into those students so that they are continuously reassured that this is a good path that they have chosen. And I think you could easily make that case, and people who don't want to listen, well, they won't. But I think this is a reasonable case to make because there is no other easy way. I mean, this is one of those damned if you do, damned if you don't, right? I mean, if your rates are going up, then it must be because the students are so good. You know, if your rates are not going up, then you must be doing something wrong. But that's not the case here. I think there's a positive intervention for all kinds of students. For example, the ones that get bored sitting in a lecture and just decide, you know, it's so much more exciting to be doing drama or to be doing, you know, accounting or whatever it is that they find interesting. Those students, when they come to these classes and on a daily basis, they are told that it's not my job to tell you whether you should be an MD or a PhD or a nurse or a journalist, but understand what is it that a scientist does and understand where they come from and then make your decisions. I think that would be a good way to do it. And I think that's a positive thing too.
Thank you. Well, just maybe a follow-up to that. So, I mean, at UCSD we have a lot of biology majors, maybe 6,000 or so, and I assume at UCLA you have a large number as well.
Yes.
Do you have any selection process early on, or is it just they just sign up?
Yeah, they just sign up. The thing is that you're talking about the earliest stage. At the earliest stage, there is a small amount of GPA requirement. I mean, we don't take somebody who is really struggling, such as 2.9 GPA or something like that. I don't remember whether the GPA is 3 or not, 3.0. So, which is not a very high bar at UCLA. Starting undergraduates at UCLA all have 3.0 GPA averages. Well, maybe not, but a lot of them have over 3.0 GPA averages. And then if someone comes and says, oh, you know, I messed up, you know, I would really like to do this, we don't say no to such people. And they have to have taken AP biology is what we ask them. But then if someone comes and says, well, I don't have AP biology, my high school never offered it, we waive that as well. So there's almost minimal amount of requirements. What is difficult is to control for the students' enthusiasm. Obviously, they heard from somebody else, this is a great class to take, and you'll learn a lot, you'll do things, you'll go, and then they come and sign up. So they are self-selecting for that.
One last question.
I think it was answered. That is, my question was, is there a feed forward on their basic biology courses? Do these students, but I think you said they're not taking Bio 1, 2, 3 because they've already had AP. Is that right?
Oh, no, at UCLA you do.
Oh, you do? Okay, so now is there an effect on their grades?
Yeah, I wish I knew how to do this. I have tried to do that. What I have tried to do is not so much Bio 1, 2, 3, which they take kind of concurrently, but to see whether or not developmental biology, for example, which is 138, which they will take a year later, whether there's any changes or not. It's not as easy to find those ideal situations. There are 5 different instructors that teach developmental biology. Some students take it in their second year, some take it in their 4th year. It's when when your control is chaotic like that, it is really difficult to see how they are doing. If you asked me, do they get A's? Yes, they get A's. But is that assessment? I don't think that's assessment. Yeah, I mean, do they get more A's than a lot of other students? That's where the problem comes in. Who am I going to choose who's going to have this motivation and in their cohort group and has taken developmental biology with one individual person, be it Volker Hartenstein or Louis RSP, or different people teach in different quarters. So this has been a difficult parameter to establish. We have tried to do it. It's not so easy. Yes?
I think that there is a different skill set required in laboratory courses than in lecture courses. And I think there are a lot of students who can ace lecture courses one after another after another. They've got straight A's in every class where they've been assessed by exams. But they get into a lab course and they flounder. They are asked to do very different kinds of things. And I was wondering what your thoughts are on that.
Yeah, no, absolutely. I mean, there's students of every kind. Then there are the ones that can really self-correct a lot, and good for them that they can self-correct. So they would do poorly in one thing, and then they will realize that they change their own habits, and this is not a science experiment. I wish it was, then everything could be done in a properly controlled manner. So yeah, there are lots of students who do extremely well in didactic courses. There too, there are ones that do well. I mean, I used to teach genetics, and we used to do a lot of problem solving. And there are some who are great problem solvers. I mean, you give them any problem that involves math, they can solve it, and then there will be the others who say, I went into biology because I thought I didn't need to learn any math, and so there's every possibility there. And what can I say? We take a group of students. This is why I started off saying that, I don't pretend to have any education background. All I can say is take a bunch of students, figure out what is best for them, and give them that. And if URM numbers go up, feel good.
Let's thank Dr. Banerjee again.
Thank you very much.